Okay, so I'm just going to go over a little bit of the sample prep things that we do in the lab. And so I've spent the majority of my time that I've been in the lab here working on sample prep and with the sample prep people that get in your samples and are the ones that deal with them. And so the first thing we wanted you to know is how to prepare your samples and to make sure that we get the because it determines your experimental success. We want to help you get the best results you can get in a timely fashion. So when we get samples in, we want to make sure that you've used HPLC or LCMS grade solvents and reagents to prep your samples. And please use 1.5 mil polypropylene tubes that are not autoclaved to send your samples in. We get a lot of things in different size tubes, and this takes our sample prep people time to move your samples into the appropriate tubes for us to be able to go ahead and process your samples.
They really love the 50 mil
Oh, yeah.
right?
Yeah, the 50 mil conicals and the 15 mil conicals are there. Please don't do that. Because we hear about it all day. Yes.
Question on that. So does this tube needs to be any kind of low bind, protein free, any special, or just a normal?
I think just the normal polypropylene tubes work fine. Yeah, just don't autoclave them. Because
Thank you.
Yeah, so you see, if they've been autoclaved, you see the detergents that are used to clean the autoclave. You see those in the tubes, and so it leads to contamination of the samples. They're clean. There's no bacteria in them, but there is polymer.
And also, please make sure to label your tubes so that they're easily read. We don't want to mix up your samples in any way. And if you use labels, make sure they're cryo resistant because they're going to go in the -80 and we don't want them to fall off. And you also want to make sure you avoid freeze thaw cycles of any of your samples. So once you have your meeting with Sam and you are ready to send your samples in, he will send you a sample list Excel file. And then if you can fill out that, this is just an example of the Excel file here. And this is one that was filled out great because you've got your sample numbers, you've got the names that are all clearly, I mean, the investigator knows what they mean, the type of sample, and then they also grouped their samples so that you have the first three are one group, the different replicates in one group, and then the other two groups. So this helps with us whenever they're doing the bioinformatics. So when it comes back out of the mass specs and doing the data analysis, it helps them if they're grouped like that. We will randomize your samples before running them on the MS. So there is that too, that you will have randomized data. Also, please be sure to tell us what kind of sample buffer you use, because that will affect our downstream processing. And if you have any notes about different coatings and stuff like that, that's great too. And these samples, they also noted that they washed them 2 times in PBS, and that was to remove the fetal bovine serum from the culture, from the tissue culture media. And then you can also see that they're clearly labeled and very easy to read. So Sam already went through this part too about how much we need. I think he had a little bit different numbers for the cell pellets. We would like to have 2 to 5 million cells. That would be great, especially for the phosphoproteomics. We do need close to 5 million. And then he also had a lower amount of plasma and serum. We would like the 50. It's just easier for our sample prep people to handle if we can get the 50. So for cultured cells, please make sure you count the cells because a lot of times you think you have more cells than you actually do. And this is an example of what a pellet for, I think that was about 2 million cells should look like. It should be about that size. Again, wash the cells with PBS just to remove any fetal bovine serum. If you could do that twice, it would get rid of that. But make sure to do it gently so you don't lyse the cells.
How many times would you estimate that we get less protein than
Less
what the
cells than
less cells than what the researcher says?
probably 60% of the time. So yeah, it kind of hinders what we do downstream. So
I have a question. So for example, we are doing the phosphorylation study. We never use PBS actually for washing cells. So can we use trace buffer to wash?
Rick?
So for phosphorylate, study phosphorylation, we normally avoiding use PBS because there's a phosphate there. So we use a trace.
Yeah, tris is fine as long as your cells can be washed with it. I mean, the important thing is rinsing them to get rid of the FBS from the media.
I see.
Would a regular saline work? Not
Um,
phosphate buffer, but just regular saline? Or would that lyse the cell? That might pop the cells.
It should be fine, low concentration, yeah. So, 100 millimolar range would be fine, anything. But the main issue is getting rid of the PBS and then actually getting rid of the solution. We get a lot of cells that, I think definition of a dry pellet in my world is different than everybody else in the world. So, for me, if I'm gonna, pull off all the supernatant after spinning down cells, there's going to be single digit microliters of liquid left. And oftentimes we'll get a tube that has 50 or 100 microliters of liquid and then a pellet, and they, call that a dry pellet. That's maybe that's
So-
dry in Arizona. I don't know, but that's not dry here.
So the purpose is really remove any contamination from the culture medium or
Yeah,
come with
That's
the
right.
FBS?
Yeah, because the albumin, especially the smaller the pellet, the bigger the issue.
Yes.
So, I've worked with clinical samples where I had 100,000 cells and there was, they were isolated in FBS. And so there was actually more protein of fetal bovine serum than the target cells of interest with 100,000 cells. And so there's just, so much mass of albumin. And so that's, getting it rinsed and rinsing it gently too, right? I mean, you want to, rinse this and spin them slowly because you don't want to lyse the cells as you're spinning them. And so it's those steps with PBS or just rinsing it's not something that's, I think, commonly done a lot of times in biological labs because you always keep your cells bathed in FBS because they stay happy, right?
Do we
making
have
sure
to
that-
mention to you that we didn't use PBS, we use trace?
Yes,
Yeah.
on that sample
Let us
list.
know. Yeah, just let us know what's in there. Yeah.
Okay.
But
Yeah,
stay
so
away
here-
from non-ionic detergents if at all possible.
Yeah.
Don't rinse them with, 1% tween.
The more we know about how you dealt with your samples before we get them, the better, because we can adjust different things. And Dennis brought this up earlier, that the batch effects can occur if you do them on different days or if you just, yep,
Yeah.
go ahead.
How long the cell pellet can stay -80?
It can probably stay for a year or so. Yeah. Yeah.
The first thing that we're going to do when we thaw them is completely denature them, right?
Mm.
So we're not doing anything viable to them. So they're probably good forever.
Thank you.
So I have a question for adherent cells. Can we wash them before we collect the pellet for it to be easier?
Yeah.
And then when we add actually the phosphatase inhibitor during in the PBS?
Yeah.
Okay. Thank you.
Okay, so for tissue samples, if you send in those, we need 10 to 100 milligrams of tissue. If you could perfuse or rinse them in saline first, that would be the best idea because you want to make sure that you can remove any of the blood or hemoglobin from the samples because that will contaminate your readings later. Try to make sure that the size and composition of your samples is uniform because we don't generally take them out and recut them and make sure that they're all the same size. So we would really like them to be the same size. flash freeze them immediately, and then store them at -80 until shipped. And that, you can leave those in there for forever too. One thing I do want to say is please make sure you don't have hair that is still attached to your sample, because that is, you're going to get keratin. I mean, it's going to show up in your samples. So, but like here, we have samples that have come in. Both of them actually have hair still attached to them. So please don't do that. Yeah, our sample prep people. So if you're sending in plasma or serum, so serum has about 60 micrograms of protein per mil or microliter. Like I said, we want 50 microliters if we can get it. It's just easier for us to handle it that way. And whenever you are prepping your plasma or serum, try to use commercially available tubes, collection tubes, and avoid any hemolysis, because that will also affect your results. So here you can see samples that were different levels of hemolysis. And so the comparison between your samples isn't as good. And then what we will do first whenever we get these samples in is run a depletion of the topmost, the 14 top abundant proteins out of there, which are albumin, fibronectin, a bunch of the antibodies, the IGs. And we want to remove those so when we run the mass spec, they aren't the ones that are showing up the most on your samples. So once Sam has his consult with you, we will make decisions about which workflow that we're going to go through.
You said there are 14 classes of proteins which are high abundant. We need to remove
We remove them.
how do you remove them?
We have a kit for the 14 most abundant, and we'll remove those from your samples, from your just from the serum and plasma protein, or
Okay, so-
serum and plasma samples.
From the mouse, from any tissue.
No, the
No.
tissues, we don't use that.
Okay. At what stage of the sample preparation do we remove them? Like before the protein, after protein extraction and before the trypsin digestion? Is it?
For the
stage-
plasma and serum, yes. Like I said, they're the only ones that we remove those proteins from.
Okay.
And it probably doesn't remove all of them either. It should do most of it, but it's just to knock down those levels of the really abundant ones. So that's not all you're seeing
Okay.
in your results. So like I said, we will, once Sam has your consult with you, we'll have a decision tree that we go through to make sure that we're doing the right workflow process. I'm just gonna,
Got another question back there.
oh, yep.
So for sending the samples, if you have beads, how do you
Mhm.
send them?
Spin them down and send them dry.
Mhm.
Yeah, but you can leave them on the beads because we'll do on bead digests of those.
Thanks.
And that's
If
actually
you
down
can-
here at the bottom.
Yeah, so if you know that you're confident that you can release your complex from the beads, then do that. Most people we find are not as good at removing the proteins from the beads. And so we get better data just digesting everything, but there's also a lot of more background, you're getting the antibody and you're getting a lot of other things that can interfere. So if you have a process that, that you've followed through and you know that you're efficiently removing, your complex from the beads, if you can do that removal step or cleavage step, by all means do it. But most of the time we get better data with direct analysis on the beads.
Go ahead.
I have another question. So, if I have samples from cell co-culture with T cells in a type of tumor cell, what would be your suggestion for preparing samples like this? And would it be possible to differentiate which protein is coming from what cell type?
Maybe. Probably the best way in determining which protein the cell type is coming from is having as one of the samples submitted an individual cell type, so that we, kind of get a map of the T cell and a map of the host cell, and then can use that to help determine where that protein is coming from. In a PDX model, it's a little easier. Sometimes you've got multiple species, and so you can do that. In a non-PDX, it is more difficult.
Okay, so like I said, we have this decision tree, and we have different protocols that we go through for different types of samples. One thing that we do for like tissue culture and tissue also is sample lysis. So we use RIPA and PIC, or protease inhibitor cocktail. Add that to the sample, then sonicate, and then centrifuge to remove all of the debris that's in there. And these were muscle tissue samples. Sometimes you want to send a lysate that you've already lysed your cells, especially for things like, I know I used to work with Staph aureus, so it's has its own lysis protocols. It doesn't really lyse with RIPA. We have to go through some other mechanical shearing and things like that to lyse our samples. If you know that your sample doesn't lyse easily, or you have to use other protocols to lyse your samples, we would like to have those lysates instead of us doing the lysis.
Or
It's
if it's
just-
really nasty, like Staph aureus, you
Yeah.
go ahead and do your own samples and vulva, anthrax, that kind of stuff.
EDTA, is that what? I don't think so, but I don't know. I haven't actually looked at the.
I don't think protease inhibitor cocktail we use has EDTA.
So once we have the lysis, we'll go ahead and do a BCA assay. I'm just going to kind of skip through some of these since it's lunchtime. And Dennis has already actually gone through after we get our protein concentrations, we will adjust all of them to hopefully 100 micrograms per sample, or we'll adjust it to whatever we need for our specific protocol, so that they're all starting at the same amount. And then each of our protocols, we go through a reduction and alkylation step. The one we most generally use at the moment is CME. And this is just goes through a little different stages of the actual process. We add our TSEP for 30 minutes at 37, our iodoacetamide for 60 minutes in room temperature in the dark. And then we will, that's added to our 100 to 200 micrograms of protein. Then we do a chloroform methanol extraction, remove the aqueous phase, which would have like your DNA and RNA and things like that in it. Add methanol, and then centrifuge and remove the supernatant, and your protein pellet will be at the bottom. And then we add TEAB and trypsin to go ahead and do the digest overnight at 47 degrees to make sure we get everything digested. And then after that, we'll go ahead and do a cleanup to remove any liquids and to remove any salts or any detergents that are left over. And then we dry down those peptides after that. So actually, Sam and Dennis have both gone through the TMT labeling, but for phosphoproteomics at the moment, this is the way we do it, and it kind of limits us to the number of samples that we can use, because the TMT labeling only has 10 different labels. So we will go through the protein extraction, like I said, reduction and alkylation, and then we use lyse trypsin to digest. Then we'll label the different your different samples, and then after you've labeled everything, you combine them, and then that's what we do the phospho enrichment on. This is the kit that we are currently using for phospho-enrichment. And like I said, it's got the titanium oxide phospho-enrichment part. And then after that, whatever flows through, you also use the iron enrichment for that. So for IPs, BioID, and things like that, any pull downs, we usually use a FASP method. And it's just basically an odd bead digest that we can do. And this just goes over the protocol here. It can't handle high percentage detergents though, so no high concentrations of SDS or anything. We are trying to move into a higher throughput sample prep options that we're using now. We just got in a new piece of equipment that we can use magnetic beads to be able to do 96 wells at a time. So we can prep a bunch of samples at a time. And these magnetic beads will bind the proteins, and then it just picks them up and moves them to different stations and washes them and everything. So it's a lot quicker and a lot less hands-on. We also have another method that's S-trap, and it's resin. That's right. This is the one, silica resin. So this, we also use this, and it's in a 96 well format also. So what we just want to make sure that we can do in sample prep is make sure your samples are all uniform and we have equal loading of protein in each sample. And these are violin plots that Dennis will go over tomorrow more in detail. And these are basically just showing that, like this one over here is showing that you have about basically the same amount of same amount of proteins in each of your samples. But if you have one that shows up like this, yeah, you have an outlier there. So this is why, like Sam was also saying, and Dennis also brought up, we want to have as many replicates as we can get for each of your samples. So if we need to throw one out because something happened to it, we can. And so this is what you would get whenever you come get your results. This is for one protein, and these are 4 different conditions. And what we want to see is little variability between your samples. Like these are all replicates of the same sample here. And we want to see this basically, so you don't have a lot of variability in your samples. This is not good. You're going to get a lot less statistical significance out of these samples. We also want to see that between your different conditions, you get a good change, fold change. So you get some significance also. So Dennis showed one of these earlier. It's a chromatogram of a normal lysate. This is about 1 microgram of helicell lysate, I think it was. But if you have contamination by blood or something like that, you are going to see something like this. I think this was hemoglobin, yeah, is what shows up for the most part, and you kind of lose your, you kind of lose all the rest of it down there at the bottom. So just as a summary, you want to limit the variability in your lab before you send us samples. Accurately measure the amount of protein in your lysates if you are sending them lysate, or if you're sending lysates to us. We're going to go ahead and measure it again. So, but it just helps if you can do that. Make sure you communicate and ask questions. We're always happy to help here. Sam is great about answering sample questions and things like that, especially. And we just want to make sure that you don't send us samples that look this variable. Like if you can see the difference between your samples, expect that we're not going to get the best results out of it.